Hello friends, welcome to BMH Learning. This video is about inoculation of microalgae. The process is divided into two main parts. We would discuss each part in detail separately. You need following equipments and materials to carry out this experiment. Conical flask, inoculation wire loop, laminar airflow cabinet, Bunsen burner, sample, alcohol, suspension media, in our case it's tap, battery plates, parafilm. The whole procedure is to be performed aseptically in laminar airflow cabinet. Let's start with the first part, agar plate streaking. So we have the grown colonies of the sample on nutrient agar. First of all, take inoculation loop. Heat it using a Bunsen burner. Keep it on heating until it gets red. Once it's red, take it away from the flame and let it cool. Now keep in mind, whenever we have to use inoculation loop, you have to repeat the process of making it red hot and then cool it. To save time, I will not do it again. With the help of cooled inoculation loop, carefully pick a well-isolated colony from sample agar petri plate. Right away transfer the picked colony on the loop to another fresh petri plate containing solidified agar. Using the same sterilized wire loop, aseptically make parallel streaks of the suspension on the agar. You can notice there are 16 streaks. So that means we have made 4 sets of 4. This way the whole surface of the agar plate is used. After this, cover the petri plate and then seal it using parafilm. Place the plate in inverted position at room temperature and light on conditions. Let the plate incubate for at least 3 days for proper growth of the microalgae. Second part is culture plate to suspension media. First of all, we have the streaked plate having grown sample. Let's remove the parafilm from the petri dish. You can observe there is clear growth of microalgae. Now, aseptically using the inoculation loop, pick a well-isolated colony from the streaked sample petri dish. Immediately transfer it into the conical flask having tap suspension media. The suspension media should be already prepared and the mouth of the flask must be covered using aluminium foil. Before we transfer the colony into the flask, the mouth of the flask must be heat sterilized using Bunsen burner. After transferring the microalgae colony, close the mouth of flask and place the flask in orbital shaker incubator set at 140 rpm and 24 degrees centigrade. Let the microalgae grow for 5 days. Success of the experiment can be concluded from the green color of the tap media indicating the growth of microalgae. This was all. Thanks for watching.